Hello students, today we are going to read about the different steps in photosynthesis, light reaction, dark reaction, different cycles like C3 cycle, C4 cycle, that is Hatch-Select cycle or CAM cycle, or also photorespiration, which is present in our NCERT. Some factors which affect the photosynthesis, we will also read this one. Okay. In earlier video, last video, we already completed this chapter up to 11.2 point, right? So today we start from this one. Where does the photosynthesis take place? The first question is here, where does the photosynthesis take place? In NCERT it is given in earlier classes. In earlier classes we complete, in very lower classes means up to 5th we know green leaves, they make photosynthesis. Then after when you come to the higher secondary schools, then you understand that green leaves, why green leaves are green? Due to the presence of chloroplast. So this chloroplast is responsible for photosynthesis. Why? Now question is, why chloroplast is responsible for photosynthesis? Because chloroplast, it contains pigments. Which pigments? We can say photosynthetic pigments. Photosynthetic pigments means what? Chlorophyll A, chlorophyll B in green plants, because I am talking about the angiosperms only. So we have 4 different type of pigments, that is chlorophyll A, chlorophyll B, xanthophyll and carotenoids. And these pigments, they are present in our chloroplast. That what is the site of photosynthesis? We can say in whole plant, when we talking about the plant, that means all the green part of the plant, they involve in photosynthesis. All the green part of the plant, that means what? Most common it is leaves, because leaves always in green. After green, other parts. So other parts means we can consider green stems. They are involved in photosynthesis. In flower, our calyx, outermost word, that is we can say sepals. They also, because they are green, they are also a modification of leaf. They also involve in photosynthesis because they are green. Then after in fruit in unripened fruit, right? Unripened fruit, they are green in color, like mango. Unripened mango, it is green in color. After ripen, it becomes yellow in color, right? Green chilies or green tomatoes, so they are green in color when they are unripened. They are also involved in photosynthesis. That means what? We can say all the green part of the plant, they involve in photosynthesis. Most common is our leaves, that is given here. You would of course answer the green leaves or in the chloroplast based on what you earlier read in the chapter 8, right? You are definitely right. You are right. Photosynthesis does take place in green leaves of the plant, but it does so also in other green part of the plant. That means I already tell you, all the green part of the plant, they involve in photosynthesis. That is here. Can you name some other part where you think photosynthesis may occurs? I'll tell. Green stems, green calyx. Calyx means outer wall of the flower. Green, that is unripened fruits. They are also involved in the photosynthesis, right? You would recollect phylloclade, they are also involved in photosynthesis. They are modified, right? They are modifications. You would recollect the previous unit, that the mesophyll cells. Okay, when we consider leaves, so leaves in between the epidermis of the leaves, the internal structure, that is anatomy, mesophyll cells are there. Meso means what? Meso means middle, and phylls means what? Leaf. In the middle of the leaf, that is in a transverse section, outer is epidermis, it is lower epidermis, right? Abaxial and axial surface is there, and in between upper and lower epidermis, we have mesophyll cells. What is given here? Mesophyll cells in the leaves have a large number of chloroplasts, right? That's why these mesophyll cells, they are a type of parenchymatous tissue. When I tell you in plant tissue, then at that time I tell you mesophyll cells, they are also called as chlorenchyma. This is a parenchyma. They are parenchymatous cell. Why they are called chlorenchyma? Because they have chloroplast, right? So usually the chloroplast align themselves along the wall of the mesophyll cells. So mostly these chloroplasts, they align themselves along the wall. They are located nearby the wall of the mesophyll cell, such that they get optimum quality of the incident light, right? So that they can get the optimum intensity of light. Now question is given here, when do you think the chloroplast will be aligned with their flat surface parallel to the walls? That is the question. Parallel to the walls, right? So look here, for this statement. This is our, suppose that mesophyll cell is here. This one is our mesophyll cell. Another mesophyll cell is this one. Another mesophyll cell is this one, right? You can understand by this figure. This is the upper and this is the lower. This is called, these are radial walls. These are radial walls, right? When light comes from this side, when light intensity is high, when light intensity is very high, that is what happens. Then these chloroplasts, they align toward the radial walls, means just parallel to sunlight. This is sunlight and this is parallel. So this process, process called, it is called parastrophy. Then this one is called, this first, this is our second, and this one is our third, right? So first this is called P-A-R-A, parastrophy, right? Now second, this condition is called parastrophy when chloroplasts are aligned toward the radial walls, or we can say they are aligned parallel to sun, so that a least amount of light they can reach. This least amount of light, or we can say optimum amount of light reach to the chloroplast, because this time light intensity is high, so they are just away from light. Second, when the intensity of light is low, means below than optimum, then what happens? These chloroplasts, they align just, we can say perpendicular to the, at 90-degree angle, we can say perpendicular to the, or epidermis, toward the apical wall, right? So this one is called epistrophy. This one is first. Now second, this one is called epistrophy. Epistrophy condition. They are perpendicular. What is given? When would they be perpendicular to the incident light? When would they be perpendicular to the incident light? At low, right? When light intensity is low, right? And when will be aligned with their flat surface to the parallel to the wall? When light intensity is high, right? That is, light intensity is high, it is low. So when low, this is called epistrophy condition. And when moderate light is coming, this time here, this side is low light intensity, this one is high light intensity, and this is moderate light. If moderate light is there, then what happens? These chloroplasts, they are randomly present in cell. So they are randomly present in cell. Then this one is called apostrophe. This condition is called apostrophy. A-P-O-S-T-R-O-P-H. Apostrophe. Do you understand this one? When do you think the chloroplast will be aligned with their flat surface parallel to the wall? When light intensity is high, and this condition is called parastrophy, right? To protect them high light intensity, they come to the radial wall, right, just parallel to the sunlight. And when light intensity is very low, to get optimum, because light intensity is low than optimum, then what happens? They aligned perpendicular to the incident light to get the optimum light, right? This condition is called, this situation is called epistrophy. And third, when the moderate light is coming, this optimum light is coming, then they are randomly scattered to the cytoplasm. That is called apostrophe. These three conditions or three locations of chloroplast I tell you here, right? Now you have studied that the structure of chloroplast in chapter 8. Within the chloroplast, there is membranous system. So when we, in the previous chapter, cell unit of life, we already read the structure of chloroplast. In chloroplast, we know we have two. One is stroma. Stroma is, that is matrix fluid. Another is membranous structure. Membranous structure is called grana. And in grana, when we consider the grana means we have two. That is, one thylakoid stacking manner grana is there. They are called as grana, and second thylakoid grana, and another one which is present only in which we can say commissos, which is present in stroma, we can say stroma lamellae, right? It is given here. What? Within the chloroplast, there is a membranous system consisting of grana and the stroma lamellae, right? The matrix stroma lamella is what? That is grana, the stroma lamella and the matrix stroma. The matrix is called stroma and the membranous system it is called grana, right? Look here, these are chloroplast and here membranous system, this is our grana, and in between there it is stroma. This is stroma lamella, this one is grana lamella or grana, and this matrix is called stroma. It is a double membrane system, right? So this is the structure of chloroplast, right? So in grana, why it is given here? Because grana or stroma lamella, this one stroma lamella, they are responsible for light reaction, and stroma it is responsible for dark reaction. That means dark reaction occurs in stroma and light reaction occurs in grana. There is a clear division of lever within the chloroplast. Why it is given? Clear division. This is clear division of lever within the chloroplast. That means what? Stroma for dark reaction, grana for light reaction, right? In grana also we know this stroma lamella, it is for only PS. It is for only PS first, and this grana it is for PS first and PS second both. So that means what here? Within the chloroplast, what is there? There is a clear division of lever. The membrane system is responsible for trapping the light energy. That's why it is responsible for light reaction, and also for the synthesis of ATP and NADPH. What are the products of, when we consider what are the products of light reaction? So then we have, how many products are there? We can say oxygen, that is released into the atmospheric air. Second, we have ATP and NADPH. ATP and NADP, these are the products which are used in dark reaction, and they are also responsible for continue the dark reaction, right? So here it is given, what are the products given here? ATP and NADP. Who is responsible for this one? Light reaction is responsible for the formation of ATP and NADP. In stroma, enzymatic reactions synthesize sugar. Stroma is in dark reaction. In matrix, what happened? Enzymatic reactions synthesize the sugar which turns to form starch. Means Calvin cycle, after Calvin cycle and reverse glycolysis, what happens? Sugar is formed, and this is further converted into starch, because we know plant cells, the reserve food material is in the form of starch. The former set of reactions, since they are directly light driven, so they are called as a light reaction or photochemical reaction. They are called a light reaction or photochemical. Why they are called photochemical reaction? Because sunlight, that is photon, is converted into it is converted into chemical energy, modes. The latter are not directly light driven. They are not directly light driven, but are dependent on the product of light reaction, and that are product is ATP and NADPH. So this dark reaction, here we consider this one dark reaction. So dark reaction is not related with dark, means night, in the absence of sunlight. It is totally depend upon the amount of NADPH and this one second ATP, right? It will continue, this dark reaction will continue up to the presence of NADP and NADH, up to the availability of NADPH and ATP, right? Hence, to distinguish the latter, they are called as by convention as a dark reaction, right? Carbon reaction. Second is carbon reaction, because these are carbon fixation states. However, this should not be construed to mean that they occur in darkness. That is, dark reaction is not they occur in darkness, right? Because they are not dependent on dark or light. They depend on the product of light reaction. So they continue up to the availability of ATP and NADPH2, which are the product of sunlight, right? This should be not be construed to mean that they occur in darkness or that they are not light dependent. It is not, they are not light dependent. We can say that light dependent because NADPH and ATP, they are also these products, they are formed in light reactions, right? So they are indirectly depend upon light reaction. The structure of chloroplast is given here. Look, this chloroplast. In chloroplast, double membrane system. We know this double membrane system. Autonomic nerve. We have two autonomic cell organelles. One is called chloroplast and the other one is mitochondria. Here chloroplast structure is given here. Look outer membrane, outer lamellin, inner lamellin. We can see outer membrane, inner membrane. Now here stromal lamellin. This one is, and this is the grana, right? Grana thylakoids. Now here I will tell you after some time, right? Here stroma lamellin and grana lamellin. This is stroma. Stroma is matrix. These are ribosomes. Ribosome. Which type of ribosomes are there? They are ribosome. Which type of ribosome? 70S ribosome, right? And starch granules are there. Lipid droplets are there. And this also. What is this? This is the DNA. Which DNA? Circular DNA. Double stranded DS. Circular DS DNA, right? Like prokaryotic cell, circular DS DNA. RNA is also there. Which type of messenger RNA is there? Polycystonic messenger RNA. mRNA. Right. So this is the structure of chloroplast. Now look here, grana and stroma. Two things. The stroma lamellin and grana lamellin. Two things are given here, and I tell you here. Yes. This side. This is the right. And one I drop from here. These are lamellins. These are lamellins. This is This is the stacking of thylakoids, right? This one, it is called thylakoid, and it is also double membrane structure. That is thylakoids, right? Membrane structure. Now here, when we consider this one, these are pigments. We can say why I draw this. This is lumen. These are thylakoids. Look here, this one is our PS1. PS1 is present this side because this is not oppressed. This side, these are oppressed because here this one is not a free end. They are not free. So these are called oppressed. APPRESSED. Oppressed, right? Lamellae. Lamellae means membrane, right? And this side, this one is called, because it is free end, so it is called non-oppressed. Non-oppressed. What is the difference between non-oppressed? Now, can you tell me this one? This one, right? This is the stroma lamellin. These stroma lamellin. So they are oppressed or they are non-oppressed or oppressed. Which one is this one? Because both side is free, so they are called as non-oppressed. Non-oppressed. Okay. Now, what is the difference between oppressed and non-oppressed? Photosystem, which is present in oppressed or non-oppressed, they are different, right? Look here. This non-oppressed, it contains PS1. PS1 means what? Photosystem first. And oppressed part. This one is oppressed part. Here, these are, which I draw here dark. So these are called as which one? PS second. Right? So oppressed part, it contains PS second, photosystem second, or we can say LHC second, light harvesting complex second, and non-oppressed part, it contains PS first, or we can say LHC first. Do you understand? That means what here? In PS first, this is non-oppressed part or stroma lamellin. Here, NADPH reductase enzyme is absent. NADPH reductase enzyme is absent. Hence, here, but what is there? Here, only cyclic photophosphorylation occurs. This side, cyclic photophosphorylation. In stroma lamellin also, only cyclic photophosphorylation occurs. That means only ATP form, but not NADPH2. NADPH2 is formed in which PS first and PS second both are involved, and these both they are present in this case, pressed and non-pressed. Do you understand what are the site of PS first and PS second? Okay. Now come to the next point. That is how many types of pigments are involved in photosynthesis. First, how many types of pigments are there? Because here we are going to read about the angiosperm only, that is higher plants. Now we have 4 types of pigments, which is given our NCERT. That 4 types of pigments is chlorophyll A, chlorophyll B, xanthophyll and carotenoids. Right? Look here. Looking at the plant, have you ever wondered why and how there are so many shaded of green in their leaves? Even in the same plant, it is totally depend upon the amount of different type of pigments. We can look for an answer to this question by trying separate the leaf pigment of any green plant. How could we separate these pigments? In previous chapter, when I am teaching you the biomolecules, at that time I said separation of pigment is done by chromatography. We use chromatogram paper, right? So this is given here also. Separation of this leaf pigment through the paper chromatography. A chromatographic separation of the leaf pigment shows that the colors that we see in leaves is not due to a single pigment but due to the presence of 4 different pigments, and these 4 different pigments are chlorophyll A. What is the color of chlorophyll A? It is bright or blue-green color. Second, it is chlorophyll B, which is yellow-green in color. Third, we have our xanthophylls. Xanthophylls means yellow in color. And last, it is carotenoids. So, carotenoids, they are orange or yellow to yellow-orange in color, right? Some graphs is given here. Let us now see what roles various pigments play in the photosynthesis. Okay, I will tell you first. I will tell you about the pigments. Pigments in photosynthesis. How many pigments involved in photosynthesis in angiosperm? In case of angiosperm, right? Angiosperm is flowering plant, which is syllabus. How many pigments are there? Now here we have chlorophyll A. First pigment chlorophyll A. Second pigment we have chlorophyll B. Third we have xanthophylls, which is in our NCERT. And fourth we have carotenoids. Alpha carotene, beta carotene, right? These are present here. Now chlorophyll A, this chlorophyll. Chlorophyll A is called universal chlorophyll because it is present in all the autotrophs. Evenly, it is also present in cyanobacteria, right? The universal chlorophyll A is universal because it is present, and chlorophyll A is responsible also cyclic phosphorylation, right? It is universal and its chemical formula is C55H70. Now look here, H72, right? O, here O, 5N4Mg. It is the chemical formula of our chlorophyll A. Now chlorophyll B is there, right? Chlorophyll A, it is that is formed in the presence of sunlight through glycine or through glutamic acid, right? Glutamic acid is converted into chlorophyll A. Glycine it is converted into, first it is converted into which one? That is our prochlorophyll, proto, we can say here, glycine converted into first protochlorophyll. Now this protochlorophyll in the presence of sunlight and H plus ion, it is converted into chlorophyll A. So these are, we know very well, these are our amino acids. So that means what? Chlorophyll is formed with through amino acids, right? Now chlorophyll B, its chemical formula is C55H70O6N4Mg, right? Xanthophil, the xanthophil, it is the derivatives of carotenoids. Carotenoids means it is C54, it is C 40H56, right? And xanthophil, it is the derivatives of carotenoids. We can say C40H56O, right? So these are the chemical formula of different photosynthetic pigment. Now, when we consider what is the structure of chlorophyll A, so chlorophyll A, it is made up of two parts. One is called pore firing ring, right, which diameter is 15 into 15 micron, right? And another one is our phytol ring, phytol tail. Phytol tail, it is made up of C, right, C20H39OH, C20H39OH, H39OH. This is the phytol tail. This one is hydro, like as to our phospholipids, right? So these one, they are hydro. So it is not hydro, it is hydro. They are hydrophilic in nature, but the phytol tail, they are hydrophobic in nature. They are hydrophobic in nature, right? Central molecule is our Mg. And this molecule is surrounded by this N sodium, right? This is the second part, and this second ring, it is different in this one, chlorophyll B. Chlorophyll, it is different. So in chlorophyll, what is the difference between chlorophyll A and chlorophyll B? In chlorophyll A, we have methane group, but in chlorophyll B, we have CHO group. That is the main difference in case of chlorophyll and chlorophyll B, but it is not in our syllabus, so we need not go to deep very well. Okay, one, chlorophyll pigments are given. So I tell you here about some information about the different pigments, right? One thing, chlorophyll C, here chlorophyll C, it phytol tail is absent. In case of chlorophyll C, phytol tail is absent. That's why it is the smallest pigment, right? And tail is absent means what? Its formula is C35. Now H is how much? When we minus this one also, we can get the which one? This is C55, right? It is now minus 20. What is there? This one, 29 plus 40. Minus 40 means what? It becomes 20. It is not. It is C35, H58 something is there, right? And then after O6N4Mg, right? This is some confusion. You can understand. This is 39. I think it is 54, 59. So this structure is very small. Only pore firing ring is present, but the phytol tail is absent in case of chlorophyll C. This is the only one. Chlorophyll D also it has pore firing and phytol tail, right? Okay, these pigments are not. Bacterial chlorophyll, it is called bacteriochlorophyll because it is also different, because its molecular structure is also different. Its formula is also different. In chlorophyll, bacteriochlorophyll, the molecular formula is C55H74O6N4 mg. This one is bacteriochlorophyll. Bacteriochlorophyll, right? Because we know bacteria means cyanobacteria. This is present in cyanobacteria. It's near about to like to this one, chlorophyll A. Okay. So these are the different type of pigments. Okay, look here, carotenoids. Carotenoids are, that is carotene. Carotene, it is the precursor of vitamin A. It is the precursor of vitamin A. Okay, come back to the NCERT. What is given? Pigments are substances that have an ability to absorb sunlight at specific wavelength. So different type of pigment means why they different color is there. We know very well when we see some color, that means it is the result of reflection. If suppose that I see red color, that means what? Red light, the wavelength of red light, it is reflected to my eyes. When I see the blue color, it is reflected to my eyes. When I see the black color, means no light is reflected, all the light wavelength is absorbed. When I see the white light, means all the wavelength is reflected to my eyes. That is, you know, understand, please. Okay. So pigments are substances that have ability to absorb light at a specific wavelength. So can you guess which is the most evident plant pigment in the world? So the most evident plant pigment is chlorophyll A, because it is universal and it has the maximum that is capacity to absorb the light. Let us study the graph showing the ability of chlorophyll A pigment to absorb light of different wavelengths. Of course, you are familiar with the wavelength of visible spectrum, and this in physics you read this one, VIBGYOR, VIBGYOR, violet, indigo, blue, green, yellow, orange and red. This is a sequence, right? Look here, the graph is given. Absorption of light by chloroplast pigment. Chloroplast pigment, we have 4 types of pigment. Now here chlorophyll A, this blue color, it represents the chlorophyll A. In this case, we easily see that the red here, blue light and the red light. This is the most absorbed part is blue and red area. Chlorophyll B also carotenoids and chlorophyll B, right? Now rate of photosynthesis measured by oxygen released. How much oxygen is released? That is called photosynthesis rate, right? What is given here? Maximum photosynthesis place in red and blue wavelength, right? In the last one, light absorbed, wavelength of light in nanometers. Light absorbed, rate of photosynthesis, black graph, which is indicated by black color. It is rate of photosynthesis and it is the rate of absorption of light. We know here, look here, they are not overlapped. They are not overlapped. Why not overlapped? Because we know only blue, this means that is our chlorophyll A. Only chlorophyll A is not responsible for photosynthesis. Other pigment, they also absorb sunlight and then they transfer their electron to LHC first, means in chlorophyll A, right? That's why these two graphs, they are not overlapped, right? So maximum photosynthesis occur in 450 wavelengths, right? From the figure 11.3a, this is the 11.3a, can you determine the wavelength color of light at which the chlorophyll A shows the maximum absorption? It is red color. Does it show another absorption peak at any other wavelength too? Yes. In which? Blue color, right? The maximum wavelength means 450. Okay. Now look at the figure showing the wavelength at which maximum photosynthesis occurs in the plant. In which wavelength? It is red wavelength, right? Means it is 450, red wavelength. Can you see that wavelength at which there is a maximum absorption of chlorophyll A? It is red light in the blue and the red region. We can say, okay, when we consider in blue or red, means red, right? And from whole, it is blue and red, this side blue and this side 650 and this is 450. Hence, also shows the higher rate of photosynthesis, because mostly maximum absorption take place in blue and red areas. That means maximum photosynthesis take place in blue and red areas. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. A is the chief pigment, right, associated with photosynthesis. Now, here some questions given, but by looking at figure 11.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? No. No overlap. Answer is no. Why not? Because only chlorophyll A is not responsible for all the absorption of, that is all the absorption of photons and formation of photosynthesis. Because other pigment, they absorb sunlight and transfer this light to the light harvesting complex. That's why there is no overlap between absorption spectrum and the action spectrum of photosynthesis. These graphs together show that most of the photosynthesis take place in blue and red region of the spectrum, correct? Some photosynthesis does take place at the other wavelength of visible spectrum too. Other wavelengths in between blue and red. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping the light, other thylakoid pigments, which one? Here given chlorophyll B, they are accessory pigments. Chlorophyll B, xanthophyll, carotenoids, right, which are called as accessory pigment. Okay, they also absorb light and transfer the light to chlorophyll A. That's why indeed they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but they also protect chlorophyll A from photooxidation, right? Okay, now here one word is given, photooxidation, because this chlorophyll B, it is responsible for the photolysis of water. That is LH second. Here photolysis of water occurs, right? So what happens here? In photolysis, first the oxygen is removed. It is nascent newborn. We can say newborn or nascent oxygen. And nascent oxygen, its effect is poisonous for chlorophyll A because it is responsible for photooxidation. So what happens here? Now we have look carotenoids and xanthophylls. So carotenoid and xanthophylls, they are, see their formula, their structure is like this one. Okay, I draw a diagram and then you can understand how they works. Look, CH3, then double bond CH, right? Then single bond CH3, double bond CH3, then so on. Like this is the structure of, this is the same CH3, and they are also interrelated with single bond C, right? Now what happens here? They are the polymer of this ethene. When oxygen plus, I write here plus nascent oxygen. This one is called nascent oxygen, NaCENT oxygen, which is poisonous. It is not a molecular oxygen, it is a nascent oxygen, right? What happens then? They form a structure like CH3, 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 and this double bond, it binds with oxygen. They forms this oxygen, right? So when this structure is formed, now this nascent oxygen, it becomes a part of xanthophyll or it becomes a part of carotenoids. When it becomes a part of carotenoids, what happens? After then, it is released by this carotenoid or xanthophyll in the form of molecular oxygen. Means what? Carotenoid and xanthophyll, they are responsible for the formation of molecular oxygen from nascent oxygen, which is poisonous to chlorophyll, right? So nascent oxygen, it is converted into O2. O2 means oxygen gas. Oxygen gas is normal, right? So they form oxygen. This O2. That's why they are called as shield pigments or shield elements. That's why these carotenoids or xanthophyll, these pigments are also called as shield pigments, because they protect chlorophyll A from photooxidation. Do you understand this one? Okay, what is light reaction? Now next question is, what is light reaction? Light reaction or photochemical phase. Photo means photon. Photo comes from photon. Photon means light energy. Because light energy, it is also processed in the bundle form. It includes light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates, which are products that is called ATP and NADPH2. ATP, what is the full form? ATP, adenosine triphosphate, and what is the full form of NADPH? It is nicotinamide adenine dinucleotide phosphate. Correct? Several protein complexes are involved in the process. The pigments are organized into two discrete photochemicals, light harvesting complex. We use in short form LHC within the photosystem first, and also in photosystem second. So photosystem we use small, that is PS first and PS second. We have two light harvesting complex first and light harvesting complex second. Light harvesting complex, it is in photosystem first. Light harvesting complex second, it is related with photosystem second. These are named in the sequence of their discovery, right, and not in the sequence in which they function during the light reaction. Do you understand? These names first or second, it is given in the sequence of their discovery. That means what? Which one is discovered first? It is photosystem first. It is discovered first. It is not due to the sequence in which they function during the light reaction, right? The LHC are made up of hundreds of pigment molecules bounded to proteins. Each photosystem has all the pigments except one. Photosystem, it contains all the pigments, means chlorophyll B, xanthophyll, carotenoids and proteins, except one molecule of chlorophyll A. Chlorophyll A means it is the central molecule, right, which forming a light harvesting system, also called as antennae. So these 100, what is given here, 100 of pigments which are bounded to proteins, right, these are called as antennae. Antennae means which can capture the sunlight. These pigments help to make photosynthesis more efficient by absorbing different wavelength of light, because chlorophyll different wavelength, xanthophyll different color means different wavelength. These are carotenoids, these also absorb different wavelength, which is not absorbed by the chlorophyll B or chlorophyll A. They can absorb. The single chlorophyll A molecule forms the reaction center. In a single LHC, how many chlorophyll molecules are, chlorophyll A molecule are present? Only a single chlorophyll molecule A, this is single chlorophyll A is present, right, which forms the reaction center, from where electron is released. The reaction center is different in both the photosystem, right? Reaction center is different because both have different capacities. Why it is called different? Because in PS first means photosystem first, the reaction center chlorophyll A has an absorption peak at 700 nanometer. Hence, it is called P700. And another, while in PS second, it has absorption maxima, means maximum it can absorb at 680 nanometer. Hence, it is called as P680. So, P700 means LHC first, P680 means LHC second. We can say PS1, P700, PS2, P680. The structure is given here. You can understand here. Light harvesting complex is there. Photon means what? Photon means sunlight. Photon represents the sunlight. LIGST. Sunlight. Sunlight is absorbed by these. These are antennae pigments. These are antennae pigments. Antennae pigments, right? Okay, they absorb sunlight. Then these light is passed to different pigments and then come to the reaction center. What happens when the light is passed from one pigment to another pigment, means from antenna, partially antenna, then after to core particles, and then to reaction center. When it is passed from one to another, some light is spent, some energy, that we can say some light energy is spent. Then after some time, what optimum light reaches up to reaction center. That means these molecules, these that means antennae particles, they also protect the chlorophyll A from high intensity of light, right? Otherwise, photolysis may be on. So these accessory pigments, they protect this reaction center means chlorophyll A from high light intensities first, from nascent oxygen also. This reaction center, and from reaction center when electron is released, so this electron, it jump from its orbital and go outward. That means outward, what is there here? Here light, that means it is here. Suppose that here water is there. It is in lumen. In lumen, water molecules are present. So this electron, because it has energy, it uses this energy in splitting of water, breakdown of water molecule. This H2 is breakdown into H plus ion and OH minus ion, right? Hydroxide and hydrogen. And this one is called primary acceptor. It comes to the primary acceptor, and primary acceptor is our phosphorylated, which is also derived or from where? From chlorophyll A. Okay. Now electron transfer. In photosystem second, the reaction center chlorophyll A absorbs 680 nanometer, in which photosystem second reaction center means which reaction center is there? It has P680 is maximum that is maximum wavelength it can absorb. It is 680 nanometer. A absorbs 680 nanometer wavelength of red light, causing electron to become excited and jump into an orbit further from the atomic nucleus. It jumps from its orbit. These electrons are picked up by a electron acceptor. Which electron acceptor? Phosphorylated, right? Which passes them to an electron transport system. Now this is passed to electron transport system, right? Consisting of cytochromes. Cytochrome B, 6 cytochrome F, right? Blastoquinone, blastocinone, that is cyanine. This movement of electron is downhill. So first, when it jumps from its orbits, it is called uphill. Now when it transfer to the that is electron transport system, now it comes from upper to lower part. That means it is called downhill, right? In term of an oxidation reduction or redox potential scale. Redox means Re for reduction, ox means oxidation potential scale, right? The electron are not used up as they pass through the electron transport chain. They are not used, but they only pass from one to another. This is that is receptor, but are passed onto the pigment of photosystem, and ultimately these electrons through this electron transport chain, they are passed to center. Which center? PS first, right? When they reach to PS first center, what happens? Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential, right? These electrons then are moved downhill again once more. When they reach to the P700, they are also expelled out. That means they also jump from their normal orbiter. When they jump means uphill, and now when they transfer to another electron transport system, then it is called downhill, right? So then they are moved downhill again. This time to a molecule of energy rich, that is NADP is present, which is this NADP is present in stroma, right? In stroma, what happens? This NADP, it is reduced to NADPH2, right? H plus H here. This whole scheme of transfer of electron is starting from PS second, then uphill to the primary acceptor, then downhill to the electron transport chain to PS first, then one more excitation of electron is uphill, and then transfer to another acceptor, that means downhill, and finally downhill to NADP. So this NADP is reduced into NADPH2 because uphill, downhill, then uphill, and now downhill there. That is a Z structure is formed. That's why it is called the Z scheme. due to its characteristic shape, right? This shape is formed when all the carriers are placed in the sequence. All the carriers are placed in sequence on a redox potential scale. Okay, a figure is given here. You can Z scheme of life. It is given very least information is given here, but you can understand what is the uphill and downhill process. So look this one LHC. This is the LHC, right? What happens here? Either it is first or second, we can say both are LHC. This one is LHT. It is not given here, but I write here LHC first, LHC second. Because here one thing is why I am saying that this is LHC first and it is LHC second, because water splitting is given here. Water splitting occurs only in LHC second, not in LHC first. That's why here I am saying this is LHC second and this one is LHC first. What happens here? Sunlight is absorbed. Now electron is jumped from its orbits. This uphill process. Look here. Now electron, it moves uphill. This one is called uphill process. This is uphill process because jumped. Now it is the primary acceptor. This one is what? Primary acceptor. Look here. Photosystem second and photosystem first is given here. Primary acceptor, primary electron acceptor. From primary electron acceptor, it comes to the electron transport chain. This is the electron transport chain, right? In electron transport chain, what happens? ADP is converted to ATP. Now here, this electron, it is transported to LHC first. From LHC second to LHC first, or I can write here, it is PS first, or we can write P700, right? It is PS second, or I can write here, it is P680, right? This electron, it is transported this one. Now electron from here, it also jumps. When it jumps, it is called uphill. This one here, it is called this side. This one is called downhill. Look at it coming down, uphill, and now this is downhill, right? And what happens? This NADP is reduced into NADP H plus H. So this is a Z scheme, right? Okay, I tell you in another. Look here, I draw a hair diagram. This is our PS second, right? Means I can write here, it is P680. These are the accessory pigments, antennae pigments. What happens? Sunlight, this is sunlight, this one is photon, means sunlight. It is absorbed and then transport to this one and then PS second. Now PS second, what happens? Electron, this is the electron. It jumps from its orbits, it breaks. What breaks? Hydrogen, that is water, 2H2O into 4H plus ion plus 4OH ion minus, right? And what? It releases electrons, right? Okay, 4H plus 4 minus, and thus it also this 4OH, right? Minus, they forms what? They forms H2O plus O2. It is released into the atmosphere plus 4 electrons are formed, right? What happens? This electron, it is used in the photolysis of water molecule. Now the electron, this electron which is released from water, that is we can say photolysis of water, it is transported into electron transport system. In electron transport system, first here, which one is there? Here we have plastoquinone, right? Now this plastoquinone, it transport this electron to cytochrome B6. From cytochrome B6, it is transported to, right, cytochrome F. From cytochrome F, it is transported to plastocyanin. This plastocyanin, it is a copper containing molecule, right? And this plastocyanin, it transport this electron to PS first. This is the PS first. It can also that means absorb sunlight. Photon, P-H-O-T-O-N-S, photons. What happens here? This electron which is transported, it is transported from this to this side and from this side. It is also once more uphill process, right? This is our electron acceptor. This one is primary electron acceptor and this one is secondary electron acceptor. From here what happens? This electron, it is used in the reduction of NADP, right? Now H plus ion, this H plus, which one? This H plus, right? Now this H plus ion, this one is used in the reduction of 2H plus, right? The reduction of NADP. Now it forms NADPH plus H, right? NADPH2. We can write NADPH2. Also one process is there. This scheme, it is called Z scheme because this structure is look like to Z. So it is called Z scheme, right? Z scheme. Another name of this Z scheme is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. We can see this one. Non-cyclic photophosphorylation. Do you understand? This is 680, this is 700. This is P700. In non-cyclic photophosphorylation, NADP or NADPH2 is formed, right? ATP is also formed here because ATP, what happens? How the ATP is formed? In ATP, in cyclic photophosphorylation, only PS1, only PS1, this is responsible only. So what happens in cyclic photophosphorylation? Look here, this electron which is jumped from PS first, this electron, it is transported to another electron transport system, and in this system what happens? The first electron acceptor is phosphorylated. First it is transported to, that is phosphorylated. I will not write here phosphorylated. So you understand this is first is phosphorylated, right? Phytin. Then it is transported to FRS to FD. FD means what? Ferrodoxin. It is ferrodoxin, iron containing molecules. Now ferrodoxin, it comes to the plastoquinone, right? So this before plastoquinone, it is given here cytochrome B, and from plastoquinone to cytochrome B6 to cytochrome F. Now it transport to, from cytochrome F it is to plastocyanin, and plastocyanin to PS first. It return backs. These are the this is electron transport system, and these are the electron acceptors, receivers, right? Phosphorylated, then FRS, FD, ferrodoxin, then plastoquinone, right? And this plastoquinone, this one, it is a hydrogen acceptor. It is not an electron acceptor, it is a hydrogen acceptor. That's why here what happens? Here H plus ion from stroma, it comes to lumen, right? And in the process of transfer of electron, transfer of hydrogen, ATP is formed. Right? ATP is formed by using inorganic phosphate. This, how many? 4 hydrogen atoms are passed through hydrogen pump. We can see hydrogen pump, right? F0 and F1 particle, and then 1 ATP is formed, right? In earlier NCERT, it is given 2 hydrogen atom passes through this hydrogen pump, but this one is wrong. Now it is correction. It is given 4H plus ion are passed through hydrogen pump for making 1 ATP. Correct? So this one, this side, this is called cyclic photophosphorylation. Right? And it is only PS1 participate in this. Non-cyclic means both PS1 and PS2 involves non-cyclic photophosphorylation, and cyclic photophosphorylation means only PS first is involved. So that is the cyclic and non-cyclic photophosphorylation, right? Now splitting of water. You would then ask, how does PS second supply electrons continuously? So PS second, how does it supply electron continuously? So because PS, we know it does not supply its own electron, but it supply electrons which are obtained by splitting of water molecule. The electrons that were moved from photosynthesis second must be replaced, and this is achieved by the electron available due to the splitting of water. The splitting of water is associated with PS second only. Water is split into 2H plus and oxygen and electron, right? This oxygen is nascent oxygen, right? This creates oxygen, one of the net product of photosynthesis. This is the product of photosynthesis which is released into the atmosphere, and the electron needed to replace these removed from photosystem first are provided by photosystem second. So these electrons which are come from water, they replace the electron which is already leave the reaction center of PS second. 2H2O is equal to 4H plus and 4 plus 4 electron, right? We need to emphasize here that the water splitting complex is associated with PS second, which itself is physically located on the inner side membrane of thylakoid. This is very important. PS second is always located in the inner side of this one, membrane of thylakoids, and PS first, it is located outer side of the membrane of the thylakoids. Then where are the protons and oxygen formed likely to be released? In the lumen or on the outer side of the membrane? Look here, where are the protons? Protons means H plus and H plus ion. They are formed inside the lumen. Why they are formed inside the lumen? Because PS second is located inside the lumen, that is toward the inner membrane, right? So where are the protons and oxygen formed likely to be released? They are formed inside the lumen, not in the stone. In the lumen, yes, or on the outer side of the membrane. They are not in outer side of the membrane. Okay, after the formation of oxygen, oxygen is released from lumen, come to the cell, and then from cell it is goes into the atmosphere, right? Cyclic and non-cyclic photophosphorylation. Now here one question is already given, but when water is split, I tell you here why two molecules of oxygen is used in each cycle. That is here we can understand from this. Look here, I write here H2O. H2O, it is split into H plus and OH minus. Now it is correct, right? Then what happens? Look, this H, it is used in for the reduction of NADPH2. For the reduction of NADP into NADPH2, this hydrogen is used. Now this OH, where this hydroxide ion goes, OH minus plus OH minus, they forms two molecules of hydroxide ion. They forms H2O, one molecule of water, plus half O2. I write here not, we are not considering oxygen is formed here, but oxygen is not released in the form of nascent oxygen. It is released in the form of molecular oxygen, right? So here I write a half O2 plus how many electrons are there? 2 electrons. 2 electrons. But here how many water molecules are involved? Look, one hydroxide from 1H2O, 2 hydroxide from 2H2O. So 2H2O is involved in the release of 2 electrons. Do you understand this one? Right? In each cycle, 2 hydrogen molecules are involved. One is, look here, reform, and one is the splits. Okay. Now cyclic and non-cyclic photophosphorylation, that is a light reaction which we can consider. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP adenosine triphosphate, which is also considered as energy coin, carry this energy in the chemical bonds. The process through which the ATP is synthesized by cell in either in mitochondria or in chloroplast. We have two organelles in which ATP that means they can form the ATP or they can synthesize the ATP. It is mitochondria chloroplast is named phosphorylation, right? Here it is called photophosphorylation, and this side it is called which one? Chemiphosphorylation, because their side it is chemically used. Here photo means what? Sunlight is used. So these are called phosphorylation. Formation of ATP is called phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of sunlight, right? In the presence of sunlight. When the two photosystem work in a series, first PS second and then PS first, a process called non-cyclic photophosphorylation occurs. Non-cyclic means Z scheme occurs. The two photosystems are connected through an electron transport chain. As seen earlier in the Z scheme, both ATP and NADPH2 are synthesized by this kind of electron flow. Means Z scheme, it is responsible, or we can say non-cyclic photophosphorylation is responsible for the formation of ATP and NADPH2, right? This side figure is given only photosystem first. So for ATP formation, only photosystem first is responsible, and this photosystem first it is present in stroma lamnae, and non-appressed wall of thylakoid membrane. So this photosystem is present on that part. NADP reductase enzyme is also absent in stroma lamellae and non-appressed part of the thylakoids. So only cyclic photophosphorylation is obtained where stroma lamellae and where in non-appressed part of thylakoid membrane, right? When only PS first is functional, only PS first means in cyclic photophosphorylation only PS first is functional. In Z scheme, both the two PS first and PS second are functional. The electron is circulated within the photosystem and the phosphorylation occurs due to the cyclic flow of electron. Look here, this is the electron released from chlorophyll P700, that is, then come once more to chlorophyll, that is P700. Why it circulate? Because this electron is not come from water molecule. It use its own electron. So this electron is come and go means in cyclic manner, right? And PS second means what? Here another electron come from hydrolysis, or we can say photolysis of water, right? That's why it never comes to PS second. That's why it is called non-cyclic, right? When PS first is functional, the electron circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening is the stroma lamellae. Stroma lamellae is non-appressed part, right? Stroma lamellae or lamellae of Grena, right, have both PS first and PS second. The stroma lamellae membranes lack PS second because stroma lamellae no PS second is there, as well as NADP reductase enzyme is also absent in stroma lamellae. The excited electron does not pass to NADP but is cycled back to PS first complex through the electron transport chain, and this is called cyclic flow. The cyclic flow hence results only in the synthesis of ATP but not in the reduction of NADPH2. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation. Beyond 680 means P700, right? Here cyclic photophosphorylation also occurs when the light of wavelength beyond 680 is available, not 680 or below than 680. That's why only P700 is functional in case of cyclic photophosphorylation, not 680. because 680 maximum wavelength absorption, of wavelength, it is only how much? 680. So do you understand this cyclic and non-cyclic photophosphorylation? Now next point, chemiosmotic hypothesis. Let us now try understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like the respiration, the photosynthesis too, ATP synthesis is linked to development of proton gradient across the membrane, but both just in opposite form. Because in photosynthesis, proton gradient forms in grana, that is thylakoids. Toward the lumen side, that is inner side, concentration of protons is more, right? Hydrogen ions are more. That is, hydrogen move from stroma to lumen, and also water photolysis is there. Then what happens? H plus concentration is more inside the lumen. But in case of mitochondria, what happens? Concentration of hydrogen ions, it is more outside the, that is a mitochondrial matrix, that is in perimitochondrial matrix. It is more in perimitochondrial matrix, not in mitochondrial matrix. So it is just opposite direction. ATP is linked to development of proton gradient across the membrane, and this time these are the membranes of thylakoids. There is one difference though. Here the proton accumulation is toward the inside of the membrane, that is in the lumen, as I explained you earlier. In respiration, proton accumulated in the intermembrane space of mitochondria. Intermembrane space means what? Intermembrane space means that is just outer membrane between outer and inner membrane of mitochondria, right? Outer and inner membrane of mitochondria. When electron moves toward the ETS. Look here, I draw a figure and then you can understand what is the difference between these two. In that is our grana, in photosynthesis, what happens? This is our thylakoids, right? And this is our mitochondria. What happens? Here H plus ion concentration, it is concentrate, that is more than this side. Means this one is stroma. H plus concentration is more H plus concentration, and this is stroma, it is less concentration of H plus ion, this side, right? And what here? Here, this side, it is less concentration of H plus ion, and in this between, this is the in the inner that is which can be perimitochondrial space, right? Here it is high concentration, right? This side high concentration, HIGS, high concentration. There is less concentration, and just opposite to this, if there is less concentration, and this side high concentration of H plus ion. That is the difference between. That means what happens? Movement of hydrogen at that is ions, it is just opposite in case of photosynthesis and in case of respiration. From here what happens? From inner to outer, but here what happens? From outer to inner. Hydrogen moves, right? ATP, for the formation of ATP, this hydrogen from in to out. For the formation of ATP, hydrogen ions, they have to move from inside to outside in case of this one, grana. But in case of mitochondria, these protons, they have to move from outside to inside, just opposite, H plus ion, for the formation of ATP through hydrogen. These are called hydrogen pump. In this hydrogen pump, we have two protein molecules, that is called F0 and F1 particles, right? F0 means facilitate, it provides facilitate transportation. Transportation means passive, no ATP is used. But in F1, it needs ATP, and also it is the response for the formation of ATP. Okay, we will read this one, this passage. In respiration, protons accumulate in the intermembrane space of the mitochondria when electron move through the ETS. let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electron and their transport to determine the steps that causes a proton gradient to develop. Okay. The first step is when since splitting of water molecule take place on the inner side of the membrane, because water molecule inner side of the membrane is inside the lumen, because PS second is present toward the inner side, right? That's why the proton or hydrogen ion that are produced by the splitting of water accumulated within the lumen of the thylakoids. First, second, as electron moves through the photosystem, this electron when water is splitting, the electron is transported from PS second to PS first. So as the electron moves through the photosystem, photons are transported across the membrane. This happens because the primary acceptor of electron, which is located toward the outer side. Primary acceptor of electron, which one is the primary acceptor of electron? It is phosphorylated, right? Which one is? It is called, its name is phosphorylated. PHEO, phosphorylated. It is the primary acceptor of plant or electron, primary acceptor of electron, right? It is phosphorylated, primary acceptor of electron, right? Which is located toward the outer side of the membrane. Transfer it electron. Now, this electron from phosphorylated, it is transferred not to a electron carrier, not to a electron carrier. This electron is not transferred to a electron carrier. That is very important. But to a hydrogen carrier, right? Hydrogen carrier. Which one is hydrogen carrier? It is our plastoquinone. Plastoquinone is considered as hydrogen carrier, right? Now this electron from phosphorylated is transferred to plastoquinone, and this plastoquinone, it is a hydrogen carrier, right? Hence, this molecule removes a proton because it is a hydrogen carrier. It also removes a proton from the stroma. Stroma is outside the grana while transporting an electron. It transports electron, but through this transportation of electron, it also carry a electron from stroma and transport this electron from stroma to where? Lumen. When this molecule passes to its electron to the another electron carrier, another means what? This from PQ it is transported to the cytochrome B6, cytochrome F means cytochrome complex, right? Then this hydrogen is transported to lumen. This hydrogen is transported to lumen. The electron carrier on the inner side of the membrane, the proton is released into the inner side of lumen, right? Or we can say lumen side of the membrane, inside the lumen. Do you understand this one? So proton, it comes from splitting of water. Second, another proton comes from stroma to lumen. So these protons come from stroma to lumen, or due to the presence, due to the splitting of water molecule, they accumulate inside the lumen, and slowly what happens? The number of that is hydrogen ion is increased. When the number of hydrogen ion increase, what happens? Proton gradient is formed, because in stroma less number of hydrogen ions are there, and lumen more number of hydrogens are there. A proton gradient is formed, right? If H plus ion is more in lumen, what is the pH? pH decreased in the lumen. You know very well. Third step, the NADP reductase enzyme. NADP reductase enzyme, which is responsible for the formation of NADPH2, is located on the stroma side, right, of the membrane, along with the electron that come from the acceptor of electrons of PS first, right? So this cytochrome F, it transports its electron to plastocyanin. From plastocyanin, it comes to the PS first, and this PS first, look, this side, it is outer membrane, outer side of the membrane, right? PS first is located here. Now from PS first, this electron which is come to PS first, it Along with the electron that come from the acceptor of electron PS first, protons are necessary for the reduction of NADPH. Look here, this side NADP is present. Now this NADPH is reduced in NADPH2. Now this H2, 2H plus, this 2H plus, it is also used from stroma, that means 2H plus and also minus from subtract from stroma, right? That was happened in stroma, number of hydrogen ions are decreased. In lumen, number of hydrogen ions are continuously increased. That what happens? A proton gradient form, right? High concentration of H plus ion is present inside the lumen and low concentration of H, that is we can say dilute, right? So low concentration of H plus ion is present in stroma. That's why here a proton gradient is formed. Do you understand? Proton necessary for the reduction of NADPH2, form NADPH2. These protons are also removed from the stroma. So these are the three reasons why a proton gradient is formed or how a proton gradient is formed. A structure is also given here. We see this structure also, right? Why are hence within the chloroplast proton in the stroma decrease in the number, while in the lumen there is accumulation of proton. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Because H plus ion is more and more, it becomes more concentration is present in the lumen. So pH decreased, continuously decreased. In stroma, pH continuously increased, right? Why are we so interested in the proton gradient? Because why proton interested? Because we know hydrogen, that proton gradient is formed and this proton gradient is responsible for the first product, that is ATP. It is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of proton across the membrane to the stroma through the transmembrane channel of CF0. CF0 means channel F0 protein of the ATP synthase. Synthase means it is an enzyme which is responsible for the formation of ATP. The ATP synthase enzyme consists two parts. One is labeled as CF0 and the one is CF1, right? Which CF0, it is totally embedded in the thylakoid membrane. Look, CF0, it is a protein. We can say it is an intrinsic protein. Already we read in plasma membrane, two types of proteins are there, intrinsic and extrinsic. So this one is intrinsic protein which is totally embedded in the membrane, right? So this CF0, it provides facilitate transportation. Facilitate transportation means what? It is a uniport. This one is a type of uniport, means only H plus ion can move from this one and only one side, from inner to outer side. This is called uniport. Facilitate. In facilitate transportation, no ATP is used and forms a transmembrane channel that carries out facilitate diffusion of proton. Diffusion means what? Diffusion means substances they move from high concentration to low concentration, because inside the lumen high concentration of H plus ion is there. So hydrogen moves from high concentration to low concentration, that means from lumen to stroma, correct? And forms a transmembrane channel that carries out facilitate diffusion of proton across the membrane. The other portion is called CF1. CF1 is extrinsic protein which is located just outside the membrane, right? And it protrudes on outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase. That means what happens? Which makes the enzyme synthesizes several molecules of energy packed ATP, right? Chemo, that is chemiosmosis, requires a membrane. This process, that is transportation of H plus ion from lumen to stroma, this process is called chemiosmosis, right? Requires a membrane. What the requirement of this chemiosmosis? It requires a membrane. Second, a proton pump. Proton pump is CF0 and CF1. A proton gradient inside the lumen, it is high concentration of H plus ion. Outer stroma, it is low concentration of H plus ion. and ATP synthase, which is responsible for the formation of ATP. Energy is how many there? There are 4 things: membrane, proton pump, proton gradient, and ATP synthase. Energy is used to pump proton across the membrane to create a gradient or a high concentration of proton within the thylakoid lumen. ATP synthase has a channel that allows diffusion of proton back across the membrane, and this releases enough energy to activate ATP synthase enzyme that catalyze the formation of ATP, right? Along with NADPH produced by the movement of electron, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, right? Biosynthetic reactions means what? What is given? The ATP will be used immediately in the biosynthetic reaction taking place in the stroma. Biosynthetic reaction is metabolic reactions. That is, we can say this is dark reaction, maybe dark reaction, or it may be the formation of other things. Stroma responsible for fixing the CO2, which are responsible for the fixing of CO2 means Kelvin cycle, that means C3 cycle, or it may be C4 cycle, or it may be CAM pathway, right? And synthesis of sugar and the formation of sugar, right? So these are light reaction, right? We can say light reaction is called as photophosphorylation.